Hate software that has a button that you click to do something, and then it has another button that says, "Do you really want to do that?" And it's like, I don't know why they don't have another button and it says, "Are you sure? Have you thought about it? Maybe we should have 5 buttons.
Consider your options."
Yeah. Well. Yeah. Anyway, I didn't hit a certain button. Yeah. Okay. We're going live. We're live. Are we live?
We are live.
We are live. Okay, so this is about Jeffrey Tomkins and Donnie Deals and all the other chromosome fusion denying fools out there. One of their favorite things Jeffrey Tomkins found in 2017, and it's a gene called ankyrin 30BL, and that's a I think that, yeah, those are actually amino acids. And anyway, we'll get into that. But that's not important right now. First of all, we want to talk about some other stuff. I want to talk about chromosomes. Okay, and I'm going to make myself big. So these are the chromosomes that you've seen, right? When you think of a chromosome, you think of this little X thing.
That's right. Except usually it's just black and white, grayscale. These are colorful.
Oh, yeah. Yeah, these are colorful ones.
I hope my chromosomes are neon colored.
Yeah. So you see the one chromosome right here on the left and one on the right, and they pinch together at this little spot here in the middle, and it's called a centromere. And it's very important for cell division. But that isn't what chromosomes ordinarily look like.
Well, you know,
this is.
I'll let you know that you just informed me so much with the picture, because for some reason I did not make that connection between the word centromere that you use all the time and that little section, that little love knot in the middle.
Good. So we're doing it. We're doing the education thing.
That's right.
Anyway,
I did 1 point.
Yep. So here it is right here. Centromere shows three chromosomes, right? And they're duplicated, so they're called sisters or chromatids. And what happens when the cell divides is it forms these long tubules, microtubules, and they pull the chromosomes apart to different sides of the cell, and then they divide right here. So,
may I ask a possibly silly question?
Sure.
When they are joined together like that, before they form into the tubules, are they under any kind of tension? I mean, is it like a spring on, releasing and then it flies out, or is that just a totally weird picture of what's going on?
Yeah, there's no tension. They actually get pulled apart by these actin and microtubules that actually shorten up and pull them to one side.
Gotcha.
Okay.
That's the kind of question you get from a rookie like me.
Right. There's another thing called meiosis, which is different than mitosis. Meiosis is when you're forming actual germline inheritable cells, right? And that's a little different, and we don't have to get much into that, but they have like 2 different. There's the mama, and there's the mama, and there's the daddy genes, and they form a tetrad, and they get pulled apart, and then that's where the crossing over happens. But don't get too caught up in that right now, because that's not important. But that is not how genes look ordinarily. That only happens for about 0.1 1% of the time, like 0.1%, or even less. That's
You're talking
only
about
during,
the little X shape.
yeah, the X shape. Yeah,
Okay.
that's only during cell division. And here we have an illustration that shows the double-stranded DNA, which you could never see because it's too tiny. It's long. I mean, it can be really long. I forget how long. You have like 3 or 2 yards of it if you were to take all your chromosomes and put them end to end. But anyway, they get folded up around these things called histones, which are, this is an octamer, meaning there are 8 parts to the histone. And then these histones get formed into this like bead-like structure right here. And then that gets all wound together and packed into a chromosome that you can actually see under the microscope. But this is only what it looks like when the cell's dividing. What it normally looks like is this guy right here. Okay. These are basically active genes. This is where the transcription actually happens and where the DNA actually does something in its normal form in the cell. And we're going to go a little bit further into that. And let's see here if I find my right illustration. Okay, so this is a nucleus, and it's got little pores, so things can go back and forth and get out. And this is, each one of these colors is a chromosome unwound. It's not all compact like that. And it doesn't have that crossover because this, well, there should be two light blues and 2 blues and 2 pinks because they're all set up and they stick to the outside of the nucleus in some spots. And those are called laminin-associated domains. I always want to say laminin. Laminin's that cross protein that the Christians love, but lamin is something that happens at the inner membrane of the nucleus, and the chromosome, the genetic material, sticks to that in various parts. And then there's other parts on the inside, like right here where these loops are, and those are called topologically associated domains. And that's where gene transcription actually happens. So there will be some of these things that are unlooped, and some of them even unwrap a bit from the histones, and they stick out into the cell, and you have these little protein or RNA making factories that make messenger RNA out of the genes. So you see but these chromosome domains are, here's another picture. This shows you a tad there's a little body of RNA called the nucleolus, and sometimes some of the protein will or a genome will stick to that. And then this one right here is active transcription. And this one is like repressed. Some of these things are repressed. And the thing is, in every different cell you have a different set of genes that are going to be expressed. That's why you have brain cells, and then you have liver cells, and the two shall never meet, right? Because of which proteins get transcribed, that sort of thing. Now here's a better picture of, see, here's like 2 chromosome 2s, and they're showing these domains, basically these regions where the chromosome is in the nucleus. So that's what it normally looks like. And if you look at it under the microscope, you could not see that DNA because it's too skinny. So you'd have to have a hell of a powerful microscope.
It's amazing how they could store all that out using microscopy and I guess chemicals. And how do you measure the ways of things? It's just
X-rays and yeah.
It's hard to get your mind around.
Yep. Every now and then we should look at chat and see if anybody has a question. There's only there's nobody watching, so
right now we have Sydney and Dave. Don't say nobody.
Well, Sydney and Dave. Oh, Sydney's here. Okay, and Dave's here. Yeah, I don't know if they're still here though because my counter went down. Anyway, okay, so that kind of explains chromosomes, right? What they really look like, and this is something that we figured out, I think, in like the late 1990s. I'm not sure exactly when we realized that these were in territories, and there's just all kinds of complicated stuff that goes on all of these little cross-linking things and other proteins that change the way your genome expresses in a cell, and it'll change the mix of proteins that get created and RNAs that get created. So anyway, okay. Now this is our picture of chromosome 2 fusion, which is what Tomkins and Denai happened. And this happened after we split from the other apes, basically. So it was oh my God. Are we hominids or hominins? I can't remember. We need Erica.
I don't know. But I was just going to say, so before there were hominins and before there was Pan, which is the genus of the chimpanzees and bonobos, there was another genus or whatever of ape that began to diverge. One population. Thank you, Sydney. It's hominins. Diverged one from another, and from that split came hominins as well as Pan, and who knows what in between or before. But eventually, it led down to Homo sapiens as well as Pan troglodytes and Pan whatever the bonobo is. I can't remember the actual subspecies name for the bonobo.
And Erica said that she would be willing to come on the channel and explain to us about that, what we know about that common ancestor, because
that would be great.
Right now, there are a lot of people that think that common ancestor did not look much like a chimp and didn't look totally like a human, but it was an upright bipedal animal, and later on, knuckle walking evolved in the chimp and the chimp line and other ape lines. Basically,
that's an interesting idea.
It is an interesting idea. The utility of that idea is that when we look at chimps and we see their the way their cheeks or their face bulges out, that may not be what the common ancestor looked like. You always have to be agnostic about what that common ancestor looked like, because we're not entirely sure. Hominins are modern humans that are extinct ancestors. Hominids are the broader term. Okay, good. Thank you, Sydney. I always get those confused. Anyway, but we have 2 less chromosomes than the chimps do. So what the idea was is that chimp 12 and chimp 13, or 2A and 2B as we've renamed them, fused together at some point after our split with pantroglodytes or chimpanzees. And so that was a prediction.
Okay, sorry. I'd heard somewhere that was a prediction.
That was a prediction. And then we went ahead and looked at chromosome banding, and we saw, oh my God, these chromosome bands, which you can, what you do is you stain a chromosome, the ones that are all stuck together and curled up, ready to divide. You stain them, and certain areas are GC rich and others are AT rich, and they will take on the stain in different varying amounts. So it'll actually show you something about the composition of the DNA according to these bands, right? And we thought, oh my God, these bands match up perfectly. And in my talk with Dr. Dan, I explained about the chromosome fusion site and what the creationists are saying about that. And I pretty much destroyed their idea there, right? So one of our predictions were that we would find evidence of telomeres right here, right? The telomere from the chimp inside of our genome. And lo and behold, we found it exactly where we thought it would be.
May I ask a question?
Yeah.
Have you got the papers where they made the actual prediction before it was discovered?
No.
Okay.
And when it comes to predictions, they aren't all the time. You're not all the time going to find those in papers. We're going by we're going by hearsay, right?
Gotcha.
Anyway,
Scientists
yeah,
talk to one
okay.
another at conferences and
They
go,
talk to one another. Yeah.
I bet there's a telomere or something in there.
Yep.
And
then, of course,
we looked, and you were right.
And the creationists say, oh, no, it can't be a fusion site because there's a valid gene, DDX11L2, a highly functional gene that crosses the fusion site. And I spent an hour and a half on Dr. Dan's channel explaining why that is bullshit. And that's not the case. So we're not going to talk about that one today.
Okay.
We can talk more about that one later. But the interesting thing is that there was, let's see here, and this is going to be pertinent. The interesting thing is there was a bunch of, a gene, right, with the first chromosome right here, right? This is DDX11L2. So this is chromosome 1. And then there were a bunch of chromosomes, or there were 3 more chromosomes over here, 2, 3, and 4. And then right smack in the middle, we had our fusion site, right? So they were saying, oh my God, this highly functional gene crosses the fusion site. But then as I showed in later annotations, this part was not included anymore, basically. This part had it was not found anywhere near these parts, right? These parts are an actual piece of a tiny little piece of a DDX11 gene that got copied over somehow by this telomeric region, okay? And this part right here just happened to have a structure that computer program said, oh, this should get transcribed, basically, along with this, and it should go ahead and transcribe the whole gene. But we found very little evidence that transcribes, and in modern notation, we've cut this out. So there's no problem.
So it doesn't get a piece of the action. Right.
And this thing right here creates not a protein, but a long non-coding RNA that does have some, it attaches to other pieces of DNA and it does something and we don't know what it does yet. So anyway, back to the other thing. Okay, so back to that. So now the other prediction was that, okay, well, when these fuse together, we're going to have 2 centromeres, right? So one of these centromeres had to go. And there's all kinds of papers about what happens when genes fuse and you have 2 centromeres. If you have 2 centromeres, the cell cannot divide properly, cannot pull that DNA apart properly, cannot form the proper X, right? So you're going to have 2 centromeres, so one of them has to go. And there is machinery to get rid of that extra centromere and either cut it out or degrade it to the point where it no longer matters. And the centromere that ended up surviving was the one from chromosome 2A, okay, right here. And the centromere that was basically snipped out or degraded was right here. So we predicted that right here we would find a fossil centromere. Okay.
Okay.
Let's see. Now, there's another way to show that. Let's see here. Here. There. Okay. Let me get over there, up there, over there. And hopefully you can see this a little better. But here was the fossil centromere right here. Let's see, at let's see, it's just, I forget, it's around 132,693,000. Okay.
Okay.
So that was right. That was it. And we'll go back over here. Oh, back over there. So now Jeffrey Tomkins said, wait a minute. In 2017, he discovered that there's another highly functional pseudogene, which is what the L means, that crosses the fusion site.
And by functional, that means it codes for a protein?
codes for a protein and does something useful in our genome, right?
Okay, so it could still code for a protein, but that protein just be nothing.
Yeah, I think they may have found experimental traces of that protein, but his research and his evidence to back up that it's a functional gene, zero. There's nothing. There is no BL, right?
I've heard.
I
think I've heard Dr. Dan over at Creation Myths explain that sometimes they do detect a little action.
Oh yeah.
These non-coding proteins or whatever, but they're like 0.0000001% of the signal to noise ratio. It's like they're just very small amount compared to what's normal for active genes and whatever the term is. I'm going to say active gene because I have no other words to describe it.
Yeah. Hi, Robin Webster, and hi, Skeptic Philosophy, or Skeptical? Skeptic Philosophy, yeah. So we have 2 more victims. Okay. So anyway, so Tomkins found this gene that crosses the fusion site, and I'll show you why that's a problem. The most serious evolutionary problem with the idea of fossil centromere, though, is that the alleged fusion site, it's positioned inside a gene, right inside of a gene. The alleged cryptic centromere is located inside the anchor D30BL gene, and the sequence spans both intron and exon regions of the gene. In fact, the part of the alleged fossil centromere sequence that lands inside an exon actually codes for amino acids in the resulting gene's protein. The gene produces a protein that's believed to be involved in the interaction of a structural network of proteins inside the cell called the cytoskeleton in connection with receptor proteins embedded in the cell membrane. The fact that the so-called fossil or cryptic centromere is a functional region inside an important protein coding gene completely refutes the idea that it's a defunct centromere. Okay, so that's his theory. And that came out in 2020. That's his latest thing, 2020. He has another little blurb here in 2018, I think. Yeah, 2018, where he talks about it again. And he says roughly the same thing. Most serious problem, concept, Tomkins. Resulting protein, this type of ankyrin repeat protein, whatever. And then he mentions Voronin in a paper called Functional Role of Proteins Containing Ankyrin Repeats. I think they've identified something like 18,000 proteins across the animal kingdom that have ankyrin repeats. And we'll look at that a little bit in the wiki. But anyway, these proteins have to do with cell membranes and the stickiness of cell membranes and how they stick together. And he cites this paper, Voronin, as if that supports his claim that this pseudogene is functional. Voronin never mentions, surprise, surprise, Voronin do not mention anchor D30BL. They don't even mention anchor D30B. They just mention overall anchor D genes, of which there are hundreds. Okay. So absolutely no evidence that this pseudogene is functional.
So he's basically trying to slip in the idea that there's a bunch of anchor D whatever genes that are functional in their own way, but this one particular one in the site, in the area that they're talking about, the location they're talking about, which is anchor D30BL, that's never mentioned. Yeah. And in any of the papers, there's no evidence that it's active. And is there evidence that it's not?
Evidence that something is not active would be impossible.
Okay, so it's never been observed to be active. Okay.
Yeah. We just haven't found a mechanism. We haven't found anything that it does. The chances are, it might create an RNA, a long non-coding RNA that does something in the nucleus. It most certainly does something, because if something gets transcribed, it's going to do something. It's a chemical. They're all chemicals. They're all going to have some tiny effect on something. There are around, I think, 1,300 of these things, or is it 13,000? There's a lot of pseudogenes, right? And they all do something. They all have an effect on how your cell basically feels, you know, whether it has indigestion or not, you know, basically how your cell wakes up in the morning, what its attitude is. So it's really, the thing about junk drawer DNA is that junk drawer DNA is out there having some effect chemically. That effect might be no more than just heating your cell up, right?
So it's just like in the junk drawer, that one AA battery that you have in there, it will affect something under the right conditions, like maybe when it's put
right.
in a device or paired with another AA battery, and that little screwdriver will do that job on that little screw if you find a little screw to tighten.
Right.
Okay.
Right. Okay.
If
so,
I'm wrong on my analogies, let me know, because I'm just trying to.
No, that's a good analogy. Good analogy. Yeah. Yeah, and the other thing is, while it's in your drawer, it's taking up space. It might poke you in the finger if you get, you know, if you get angry and you reach into the drawer real fast. Right.
There are detrimental effects of the junk drawer.
Yeah, conflate biochemical activity with function. They have different definitions in genetics. Thank you, Sydney. Excellent. Okay. Anyway, he says this type of ankyrin repeat protein is associated with the plasma membrane of eukaryotic cells. We know that from the Voronin paper, and is implicated in the interaction of the cytoskeleton with integral membrane proteins. Voronin and Kiseleva, 2008. Kiseleva. Yeah, the fact that, he says that it's at odds with our defunct centromere. Surprise, surprise. Again, Voronin does not talk about this particular pseudogene. He talks about the hundreds of other anchor D genes that are indeed functional in the membrane. And so he lies.
Can we talk about the logical fallacy that that he's trying to introduce here? Is that simply because some in a class do something, that doesn't mean that all in a class do it. And
exactly.
you'll have to have evidence that something, this one particular 30BL is doing something, and nobody's ever found that.
Thank you so much, Scott. This is why I need you. Anyway.
I guess that's a fallacy of composition.
Yeah, I think so. We have to have our fallacy expert to tell us what.
Where is our fallacy expert?
Where's our fallacy expert? This is not showing up. Let me see here. Do I have to make this smaller? Yeah, I do. Let me see here. Okay, so let me adjust this. Okay, this is I think this is from Tomkins' paper. Yeah, it is. It's from one of Tomkins' papers. The alleged cryptic centromere site is a key sequence. Okay, you see over here that we have the fossil centromere site that we found, and I'll tell you why that is. There's alphoid Hoare repeats, higher order repeats, basically. And there are some lines and signs in here, but there are no genes basically here. And we have this anchor 3D, anchor RD, 30BL, which I think is right here. And it starts out over here with one exon or two exons on one side of the fossil centromere, and ends up over here with a bunch of exons. I think 2, 2, 6 are over here on the left-hand side of that. And this whole cryptic centromere thing covers about 114,000 base pairs. This is the whole anchor. Yeah. Oh no. The blue on top. Okay, you have one exon. It's read in the reverse direction, so it reads backwards. So the first exon is on the right, and it reads backwards. And you see that big long blue line, and then boom, way over here, we have a couple of exons. So we have a little cluster of exons here, and just like DDX11, on the other side of the predicted site, we have one little exon, okay, that, as it turns out, doesn't code for anything. In fact,
Wah, wah, wah.
yeah. Anyway. Okay, so then let's see, debunking the debunkers. He's got another paper here. By the way, my description is rich with everything that I'm talking about here, and I do have enough for you to find it on Google. And why do I have that repeated again? I don't know. I'm not sure.
Now,
Anyway,
the debunking the debunkers, was that his response to like
yeah,
Dr. Dan and Erica?
right. And people that are calling it a pseudogene. And he's telling them, oh, no, it can't be a pseudogene. So here again, he mentions the same thing. He mentions Voronin and Kiseleva. Chr2 implicate in embryogenesis and a regulation of many intercellular processes, doing his compositional fallacy, that because there are hundreds of genes with an anchor D repeat in them.
You need to wake up the converted man and get him to comment on the fallacy.
Yeah, I know. Yeah. Anyway, so he does it again. Okay. Now, the thing is that he's got a problem. Evolution has a problem because, okay, if there was an original anchor D30B gene, right, that got copied into this site somehow in the human after we split from the chimp, then why would one exon end up over here and 2 to 5 end up over here? So how is this gene copied into that site? So this would have been a killer bit of falsification of chromosome fusion had it turned out to be correct. Okay, so that's the important thing. You see the original gene here gets split in half. Well, that doesn't happen. That could happen in some weird circumstance, but we'd have a hell of a time supporting that, right? So here's the human fossil centromere right here. Okay. Now, I want to switch over and show you something else. Okay. Now, this is the entire anchor D 30BL gene that I have got from the latest, greatest telomere to telomere long read DNA, which is really reliable. And up at the top here, you can see you can see uppercase letters. That is the first exon, last exon. This one is in the forward direction. This one is in the reverse direction. So it shows you the first exon because it's reading backwards up here, right? Okay. But we're going to look at the forward. Now, when you look at this anchor, somewhere in this anchor D30 gene, there's a centromere, okay? And I'm still scrolling down, and we're only looking at 110,000 base pairs, right? We're scrolling down, and let's see, do I have them? Let me see if I got what this one is. This one is this one. Oh no, okay, no, that's the whole thing. Anyway, when you go down, oh my goodness, oh no. Okay, when you go all the way down to the bottom of this one, you'll find the first exon, or you'll find this cluster of exons. You'll see that this is exon 6, I think. This is 5. This is, you can see where the uppercase ones are just exons. All this other stuff, this lowercase stuff, that's all intron. That all gets snipped out. That's all junk or junk drawer stuff, right? Doesn't really do anything. So anyway, but you can't, do you see the fossil centromere? No, I don't see it. Do you see it?
Nope.
I don't see it. Okay.
But, okay, getting back to the fallacy, I went ahead and invoked Copilot's AI and asked the question, what form of fallacy is this? Copilot says it's a hasty generalization with a category error and a faulty analogy. And then I said, well, is it a compositional fallacy? And it said, yes, it is. So I don't know. It could be among those. I don't trust an AI to not tell me what I want to hear. I wanted to hear composition fallacy because that's what I guessed, or that's what I came up with, but it could be a hasty generalization.
Yep. So anyway, I'm showing you all the DNA that goes into the anchor D BL30 or anchor D BL30L gene, the pseudogene, right? And you can't see it because this is just one line right here. I got it all on one line. So what I did is I split it. Okay, we know that centromeres have these higher order repeats of this 171 base pair pattern that gets repeated over and over and over again in a centromere, right? For about 1 million base pairs in a centromere. And that makes that little pinch part that, you know, hooks up and makes the X in the genes, right? When they divide. So what I did is I thought, oh, well, I'll do 171 per line. And up the top here, okay, this is the forward read. So we're going to be reading exon 5 first right here, right? So this is the last exon. This is exon 4. You can see how close exon 4 is. Exon 3, exon 2, right? But okay, it looks like junk. I mean, there's no pattern you can see in here, okay, of any great pattern. Let me see, where do I have it? Why? Oh, but then 132. Okay, well, look at this. Look at that. So can you see the pattern right here? Scott, are you with me, Scott?
Oh yeah, I've been talking and talking. It's I'm muted. I do see a pattern. Actually, I do. I never thought I would. You're talking about here in the middle section?
Yep, right here. Yeah. There's, look how all these things line up. Okay.
It's amazing, the human brain, because I have no way to read each individual letter, but I can still see the pattern.
Yes.
It's wild.
Yeah. And it goes on. And this is the fossil centromere site. And it's right smack in the middle of that gene with an intron, and it's still going on. Look how big it is. And it just keeps going. And of course, it gets a little screwed up, and it keeps going. And here's pretty much the end of it. So the end of the fossil, right? Okay, but we're in the intron between the first exon and the second exon. So we have a problem for evolution. Oh, the pattern starts again down here and keeps going further, basically. So there's more. And yeah, okay, this is the true end of the fossil. So you can see how many repeats there were. There were quite a few. And now we're going to end up at the first exon, which crosses that fusion site. So evolution still has a problem. Now, if we go out here in Pantro, okay, and we take a look at the centromere, okay, we can see this is Pantro 171. We can also see that there's a pattern. It's a little bit skewed, okay, but that looks, you can see that these things kind of line up, right? There's all these different patterns, and this is the actual centromere. The old fossils and the centromere they got killed basically actually looks like this in the current chimp build of the genome. Now, we didn't have any of this stuff available in earlier reads at all. They just had big blank regions here where they basically made shit up. So you can see that the centromere is really big, and I'm only about one. I'm not even a fourth down yet. Basically, you can see the centromere goes on and on and on. It goes on for about 1.1 billion base pairs of DNA, the actual centromere, right? The fossil centromere is 110,000. So it's 10% of this, right? This is the chimp, okay? And here's the human fossil again. And with all my exons. Okay. So you got that? So you can actually see centromeres, and we can see that we have a fossil centromere in here. This does not exist anywhere else in the genome, only in chromosome 2 that you'll find this kind of perfect repeat pattern that goes on and on and on and shows this pattern. So that was our prediction, that we'd find a fossil centromere site, but we still have a problem, okay? There's still a gene across that thing. So
and
that's
before we go address that, can we can we look at Robin Webster's question? It's a good question.
Oh,
One of the best questions. I mean, we have examples of fusion in other animals, correct? Do they accept those? In other words, creationists, other than creationists? Oh, and this was predicted. So in other words, she's asking if this is true for other species.
they won't tell you. They won't respond to it. You pointed out to them, like in pigs, we have examples of these fusions. We have examples between horses. I think different kinds of horses have different numbers of chromosomes. Different mice in Germany. Like there's populations of mice in Germany that have 48 chromosomes and some have 19. And these are all things that were one created kind, right? And so they have examples of chromosome fusion, but they'll say, oh, no, it's different because we have this functional gene crossing the chromosome fusion site.
Now, what kind of fallacy is that?
Ignorance? Ignorance? Ignore it?
Special pleading. No, that's different.
Special pleading, yeah, okay.
Unless, of course, you can come up with a legitimate reason why, no, that doesn't belong in this class of things, or this phenomenon is not quite the same with the same implications. But that's a special pleading fallacy. So fallacies all the way down. Here's a skeptic philosophy, getting back to the fallacy of composition. Assuming that something is true of part of a whole must also be true of the whole. So, and the correction to, I guess, their fallacy is that many of those anchor ANKRD genes do function XYZ, but not all of them. And there needs to be indications that anchor 3D11, whatever you call it, performs that function.
Yep.
And there is none.
Okay, so what I did is I went ahead and I took that first exon, basically, which is over here at the very top, anchor BLK 30. Let me go to the top. Okay, no, that's the bottom. I took this first exon down here. No, I'm sorry. This one up here. The one that's way on the other side of the fusion site. I keep forgetting where I'm at. I got these things forward and backwards, and that really is tough on my brain, just like time zones are. Okay, so the first exon right here, I took this sequence right here, okay, this whole sequence, and I went ahead and I thought, okay, is that in the chimp anywhere? And I did a BLAST, which is a basic local alignment search tool, okay, a BLAST, and looked for it in the brand new, latest, greatest chimp build, and guess what? I found it right here. I found it in this purple line right here on the right side of the chimp centromere for chromosome 2B.
So when you find examples like this, do you just like jump up and shout, Eureka?
Exactly, I did, yeah. Okay. Well, it wasn't Eureka yet. The other thing is I found the rest of the gene, okay, over here. Wow. In the chimp on the left side.
That's a lot
of
I
found the first exon. Yeah, that's a Bayhude centromere. Okay. And so I found that first gene, that first exon, which has that, for some reason, there's just a lot of room. And then you get to the last exon. Well, in the human, this is 100,000 base pairs long. But in the chimp, it's 1,100,000 base pairs long. So what in the hell kind of gene basically would start here and go all through the centromere and then keep, finish its translation over here on the end?
So to kind of summarize it, before there were chimps, before there were humans, probably before there were hominins, there was a ape, primate, maybe a hominid, but not a hominin, that had the full gene, all 1,100,000 of that gene. And then some split happened, and the genes on the human side got cut out, or the whatever got cut out, resulting in future Homo sapiens having the shortened version, and the chimpanzee, which had not existed yet, but went down a separate evolutionary path, and it took on, well, it kept the whole thing. Okay.
Yeah.
Sorry.
Okay. But the thing is, this first exon, as it turns out, has nothing to do with the rest of the gene. It was just a false read, basically.
Oh, man. And here I wasted all that verbiage on it.
Yep. And now if you look at this part here where this actual pseudogene is, you'll find it in the gorilla, and you'll find it in the chimp, and you'll find it in the human, okay? In the same places. In the gorilla, it was in a different chromosome. I think it was 810 or something. But then if you look at the actual anchor D and KRD30B gene, it's right by the centromere in chromosome 18 in humans and chimps and gorillas. Okay, so you'll actually find it. You'll find the whole gene, but we'll show you that later because there's more evidence here. But anyway, the thing that I found is that, okay, you get a hit on one side of the centromere in the chimp, you get a hit on the other side. And I thought, oh my God, I discovered something brand new. Here is the human. I think this is human. So this is the fossil centromere right here at 33.4. And here's the chimp anchor D BL gene. Oh, no, wait a minute. No, I think this is the chimp. They actually annotated it in the chimp. So if you look now, when Tomkins looked in 2017, there was no anchor D30BL gene in the chimp. Now it's there. Somebody annotated it. But looky here. They don't have that exon on the other side of the centromere. There is no exon here. The gene starts here on the left side, or the early part of the chromosome, the left side of the chromosome, and only runs for that long. And that is the whole chimp anchor D gene. So then my eureka turned into oh shit, somebody else found this. Why the hell didn't they tell Tomkins? Why didn't they tell Tomkins? You're wrong. It's in the chimp. It's right there.
Somebody probably doesn't talk to Tomkins because Tomkins is a wackadoodle.
Do we know how and why this would happen? Is it just copying error? Robin, this thing happens everywhere in the genome. There's tons of pseudogenes. There's tons of little fragments. Genes get pieces of the genome get deleted. They get inserted. Large pieces get copied. Viruses come in. There's overlaps. There's a ton of different mutation devices that we've identified that will cause this constant shuffling of genes. And what happens is if a valid gene gets cut in half or hurt somehow, then the organism dies before it ever gets out of its mama, before it ever becomes a fertilized zygote. And so natural selection takes care of everything except the parts that don't care, right? Yeah, maybe he just ignored it. Well, he's been silent since 2020 about chromosome 2 fusion. So maybe the chances are that these creationists aren't looking at the latest, greatest genomes because it kind of scares them. But anyway, so I have another full picture here where you show the first exon, okay? And this is, let me see here, is this Pantro? Yep, okay, so this assembly is Pantro 3 version 2.0, which is the current one that really covers the whole genome of the chimp. And we have the chimp centromere, 1 million base pairs. We have the exon way over here. And then over here, we have the annotated, that little green thing right there, that you can barely see it. That is the annotated anchor D 30BL gene in the chimp. So it's there. Okay. And it's on the left side. It's pretty far. Now, what's interesting is that in the human, okay, everything from here to here, I've been able to find on the right side of the chimp. When I blast human genes against the chimp, I find all of this stuff, right? And I find the fossil centromere part of it. But then 1 million base pairs are cut out of the human. And that's how the centromere repaired itself. And I got into a fight with ChatGPT over this because it said, no, it probably degraded. And I said, bullshit. There's evidence that 1 million base pairs got snipped out. And the way that happens is, and this is for Robin too, when you have these common areas like this, right? When things are common, when the gene lines, one gene lines up against, basically it duplicates and there's some sort of a break, how does it know to repair the gene, which one of these to use? So what will happen is this one will get overlapped all the way over to this one, right? And it'll chop out a huge section of it. So when you have these repeats, when you have areas that are highly similar, frequently, if there's a DNA damage, it will try to put them back together. It'll try to match them up, and it'll end up snipping out a whole huge chunk. Sometimes it actually causes more of a duplication. Sometimes it just deletes a whole huge chunk. So does that make sense, Scott? Scott, are you muted?
Yeah, you have to give me some time to find the mute button because I lose it every time.
I know. So, boomer. Okay, boomer.
I'm a boomer. I can't zero in on those things. But yeah, okay. It makes sense. And it's kind of like, you know how like when you're having to repair maybe your car because a rat got up under there and chewed off your headlight wires, just as a hypothetical.
Yeah.
When splice the wires back together, sometimes you have to clip out the little damaged section of wire in order to splice it back together. And of course, what was happening here was there's a redundancy. So the way that the gene eliminates redundancy is, I guess, to cut out part of it to so it's not redundant anymore because you have a partial non-function. I hate the word function because I don't know how I'm supposed to use it, but you have the non-working remnant, and then you have the working one that was a duplicate.
Yeah, function is a human idea. It's a human word because we think in terms of function and tools. There's nothing in biology that should be called a function. I guess biology is just chemistry.
That section no longer behaves chemically like it did before.
Right. So then I'm not sure if I did this in the human or the chimp, but it doesn't matter. But I went out, I think it's a human, and I went and searched for the anchor D30B gene, the actual gene, the real one that actually does what Tomkins claims this pseudogene does, you know, that has an effect on cell membranes. And I found it, and guess where I found it? I found it by a fucking centromere in chromosome 18. This is a centromere right here. You see that little pink? Okay, it's right there. So when somewhere along the line before gorillas split with chimps and humans, there was an accident basically, and a pseudogene got copied into the chromosome 2 area right by the centromere. Because the thing is, when it tries to repair, what it does, Scott, is it tries to match them up and say, oh, okay, we want to line them up one for one. But if there's a lot of repeats, oftentimes it doesn't know exactly which repeat to line up with which repeat, and you'll end up with a deletion or basically a duplication. Okay, so it tries to find homology, and if it gets fucked up because of repeats, it won't find it. So anyway,
interesting.
What else do I have here? This is the actual picture of the gene. So this is in humans. Some reads basically get that first exon. Some of the older reads found that first exon to be transcribed into the mRNA. Whether they did that experimentally or if they did it by a computer model, it's hard to say. It's an NR, so you would think it was looked at by somebody meaning it's an error. But then we have all kinds of new reads of that same gene, basically, that say, no, this is the fossil centromere site, right? And they're saying, no, there is no exon over here, basically. We can't find it. And there are good reasons for believing that that exon has nothing to do with anything, which I'll show you in a minute. This is this is a blown up picture of that same diagram. So you can see the, you can see the ones on the left, right? There's a bunch of evidence here. NR means that somebody looked at it. It's a ref-seq gene. It's something that somebody's looked at. NM means that it was modeled by a computer, basically. And XR and XM, I forgot what they mean, but roughly the same thing. So anyway, let's see. So this is the cytogene. Okay, this is chromosome 18 again, showing you where the Chr2 gene, 30BL gene is. Okay, so let's go look at the pseudogene. That's what the protein looks like, right? Okay, this is the actual protein, and you can see the Chr2 repeats, which are these alpha helices here. These are the repeats, and this forms kind of a little solenoid thing that kind of grabs onto some other protein. So this is, yeah, the blue ribbons, yeah. Yeah, those are alpha, basically alpha coils. DNA just tends to form that structure very easily.
Beta coils up in my DNA. I'm totally
there are some beta ones, beta parallel sheets.
Hush your mouth.
Yeah, no. Okay, but let's go look at the real gene. Okay, so I want to make that the right size again.
The non-pseudo gene, the real thing.
Yeah, here's the real gene.
Classic of genes.
This is the Chr2 30B that actually has function. You see the Chr2 repeats right here?
I see them.
Okay, we'll
blow
them up.
The ribbons, yeah.
Yeah, I'll blow them up. But look how much more there is to the gene. Okay, that is not in the pseudogene.
Gotcha.
The pseudogene is 242. I think, I'm guessing alpha.
So even even on the left, those yellow pasta curly Qs and the light blue and the dark blue ones, that's part of this gene, and that's cut out of that chromosome 2 centromere thing.
Yep.
Okay.
Yeah, none of that's in there. Okay, it's all cut out. Basically.
Wow, it's good to be able to put this in context.
Yep.
In context of a working gene.
Yeah. And then let's see. Okay, nucleotide sequence. I don't know why I put that in there. I probably shouldn't have.
It's impressive.
Oh, here's the pseudogene, 258 amino acids. Here's the sequence they found in old reads. In the new reads, we got a problem. I'll show you the problem later. But the old reads, they found, okay, ATG is methionine. That's that M right there. So that is the start codon. A gene has to find a start codon, or the ribosome finds a start codon, and then it starts creating the protein after that. So it found that start codon, it went along, and it created this putative gene, and a very small part of that is the alphoid repeats, or not alphoid, the Chr2 repeats. Basically, they create the solenoid. Okay. So this is Chr2 30BL. This is, again, let's see. Okay, this is exon 1. So now we're looking at it backwards. This is actually read right to left. Here's exon 1. Here's a possible exon 2. And this is where the fossil centromere is. And those dark blue regions are where it actually starts coding for protein. So it doesn't actually code for that exon at all. It's just sort of garbage that gets capped and thrown away, basically. Doesn't do anything. Okay. Now, what I did is I lined up the pseudogene with the actual gene, and the actual gene runs way down off the page because the actual gene is like 1,300 amino acids. The pseudogene only has 252 or something like that. And okay, the beginning doesn't line up at all. These dashes mean that there's no homology. I did this in Clustalo Omega, which basically does an alignment for you. It starts right here, and we have some good homology right here. This is why they call it a pseudogene. Those stars mean that it does line up. So that many amino acids line up, basically, and then from here on, it skips a big region, and then that lines up, and then all that doesn't line up. So there's no, basically, homology, the very weak homology. So this is why they call it a pseudogene, because in all of the genome, they found something that was a significant statistical match, basically right here, that said, oh yeah, that looks like an old ancient accident that got copied in and then mutated all over the place. We had a big insertion here, and we had deletions and all sorts of things. Okay. That's all pseudogenes happen. Okay. Ready? And then, of course, this is the Chr2 gene and what it does. And that just tells you what it does. And I believe that's pretty much what I got. But I wasn't done yet. I wasn't done. I went ahead and I did a whole bunch of stuff. I went and I tried to, here's my alignment. And then what I did is I went and I tried to translate. Okay, I took the current genome, the really good, reliable genome, and I tried to translate that into a protein. I skipped the first exon, which had all kinds of problems, didn't do anything. Started right here with my start codon, M, and read across. Basically, I should make this a little bigger, shouldn't I? Should I make this bigger? Is that going to screw things up? I don't think so. Okay, a little bigger for you. So I found the first ATG and I started translating. And what I ran into is protein. Stop codon. It's going to, all these stars here are stop codons. They aren't going to work. That isn't going to translate it into a protein. There's two stop codons in a row. So then I shifted it over and I went and looked for the second one. I think, let's see, where did I do this? Down here.
Okay, so just so people won't feel so like everything's over their head. When you say stop codon, you're basically talking about the equivalent of like when Morse code, when they used to do telegraph in Morse code. In the message there would be dot, 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 and somebody would say stop because that's the end of the sentence. That's the period.
And they would stop.
Like, go no further. So that's all a stop codon is. So when you hear that word, don't be thinking technical jargon. It's just the chemical command, so to speak, to stop doing what you've been doing.
Yep. And let's see. I got to get rid of regular expressions. I just want to try. So I tried in a number of different ways to translate that protein and come up with something that looked like the old 2004 protein, the pseudogene that they think they found. And I just could not do it. I couldn't find anything. Oh, let's see here. Where's a star? Hang on a second. Star. Okay. So search. Let's see. Repeat operator. Okay, I'm sorry. I got regular expressions on. And I'm going to try to find all. Okay. So I tried the first ATG, and I got stop codons all over the place. You see all these little red marks. I tried the second ATG. I got stop codons. I tried the third ATG, the fourth, the fifth one. Tried it, and there was no way to even get a match with what they found in 2004. So anyway, didn't happen. And all of this stuff is just the work I did to find the exons and to go ahead and this is the actual whole goddamn gene again. Let's see, where do you end? Where do you end? I'm trying to find it. I think it's at 2000. Oh, sorry. Okay. Then let's see. I read the chimp and I read the human and I found the locations of the exons. And then again, I cut out the exons. And somewhere down here, I have, let's see here. Let me turn that off. View. I don't want to look at word right.
Apparently you said they a second ago, and Robin wants to know who is they. Tomkins?
No. Researchers that worked on the genomes. The people that annotated the original, when there was all that hullabaloo in the early 2000s about we've read the genome, there were a whole bunch of people that went to work. They gathered all kinds of information from every bit of research they could, and they started annotating genes and putting them together. And they did it with computers, and they did it with a little bit of hard work. And we came up with ideas about what genes we have, but they were just ideas. And when you read the genome back then, what we all needed to tell ourselves is this is not completely accurate. We have to take all of this with a grain of salt, sort of thing. What else do I have? Too random. Oh, these are the actual alignments that I did, basically. Let me see here. So let's see, pantroglodyte. So I did an alignment. So this is pantro down here. This is the gene from the sequence from the human, basically. And this one was pantro. Let's see, which one am I looking at? Pantro, related information, idea. Okay, shit. Boy, 347 to 348. I'm not sure if this is the first one or the last one. But now, okay, you can see where there's little gaps right here. That's where it doesn't line up between humans and chimps. So when you talk about human and chimp similarity, these little gaps are places where we're different. These big long strings of vertical lines are where we're identical to the human. Even our pseudogenes match up. Even genes that don't have any use at all, basically. Why didn't I write down the exon? Oh my God, I didn't write down the exon. Anyway, so this is another exon that I did. So I did the first exon and I did the last exon, and I found them crossing the centromere in the chimp, in the human, and this proves, this is strong, the strongest evidence that we have a common ancestor with chimps and that there was a chromosome fusion. So this is extremely strong evidence. So Tomkins stumbled on something here. Let's see. Is the gap a place where the chimp just has, no, not less DNA. It means that it doesn't match. So if there was, I think they would have put a dash there. Like, okay, if there was an area that didn't match, I can show you that over here. Where, oh, I forgot where it is. Anyway, the gap means there's a C in the human and a T in the chimp. See that right there? C and a T. So those don't match exactly. All these other ones, we got AAAA, we got AAA. Those match, okay? Now, if there was a gap where there was a deletion or an insertion, one of these would have dashes in it. It would look like, let's see, Control O. It would look like dash, 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 like that. I don't know why that didn't work. Anyway, but there are no deletions and insertions in these two examples. Does that answer the question? Okay.
We have confirmation. He says, "Oh, I see that now." Thank you, Skeptic Philosophy, for your question. And Robin Webster for asking so many questions as well. We appreciate that.
Yep. And let's see, we could do, let's see what else. I think we pretty much covered it. I've listed the genomes where I got all this information in the chat. UCSC, University of California, Santa Clara. I think that's how I said that right. And the other Santa Clara. So I think it's Santa Clara. I don't know. Maybe it's Santa Clara. I'm not sure. Oh, maybe it's something else. Who knows? UCSC genome browser. That's all I care about. I listed those in my
California, Al. It's way out there in California.
Yeah. University of California. University Sandy. Oh, I don't. Who cares? And then NCBI is the other one, and I listed that, and there's some other resources that I put in. Let's see. Answers. That's why pairwise alignment measure. Yeah. Well. Okay, if you look at the similarity of what I did there, it's listed as 100%, basically. So let's see, where is it here? Somewhere they have a percentage. Can you spot it? 0 to 342, 0. Okay, 99%. We're 99% identical right here. And the first one I think was 98%. So in the chimp and the human, you'll constantly find when you align areas, and yeah, the reason why they say 84% is because they count every deletion as a mutation, and that's not the case. If you had a big string of deletion or copy or deletion or duplication, that would be one mutation, basically. So, and that happens in areas that aren't conserved at all. This one is pretty highly conserved, which, but not totally. And what we've seen is that this protein, it no longer codes for a protein because of those little stars I showed you before. I'm starting to lose my brain a little bit.
Well, that's good because my dog is getting more and more upset that I won't.
No, Robin, I tried to explain it so well.
Well, maybe I bet if you'll rewatch the video.
If you watch from the beginning,
basically it's just a big text file that you're comparing, but then you can comment on the chemical reality, of that big text file, and you're just comparing where things line up with a chimp and where things don't, and looking at the patterns around it to see why it doesn't line up and what happens because it doesn't line up.
So I'm asking the question in chat. Robin, were you watching from the beginning? Because I think I set it up pretty well in the beginning. If you haven't, yeah, okay, you started late. So if you go watch the beginning, I explain the X chromosomes and chromosome areas, and then I go on to explain why Tomkins thought this was a problem for evolution, and I do it.
This is something that just for a person that's not trained in this, and just educated with hours and hours of college and graduate level education, you just have to go over it and over it and over it and over it until it clicks. There's just no way to avoid that. Yep. And believe me, it's barely clicking. It's barely a tap for me.
Well, I'm hoping that I did a good job of explaining why this would have been a problem for evolution. And anyway, that's about all I got. So the bottom line is the gene is in the chimp, and the exons are in the same place in the chimp, right around the centromere. A big chunk of the centromere got cut out after the fusion, probably within the same generation that the fusion happened. There was a repair mechanism that cut out that extra centromere. That's why they were able to reproduce and go forward. And Tomkins' idea of anchor D30BL is completely out of the question. I mean, has nothing to offer at all. Tomkins is dead fucking wrong. That's the bottom line. Okay.
But of course, he won't listen to you. I mean, even if somebody showed him this video, I imagine like in Clockwork Orange style, propping his, pinning his eyelids open and making him watch this video, he would still go, this guy's not educated. he doesn't know what I know. You know, forget him.
Yeah. Yeah, what he'll do is he'll go along here right here and he'll show you all these expression. Basically, this is all epigenetic information, all these lines down here. And you can see right smack in the middle of the human gene, there's all sorts of epigenetic information that says that something may be expressed. Yet we don't have any known genes here at all. It's one big intron. And if you look at the chimpanzee, we have the same thing, basically. Here's the chimp. Okay, here's a few areas where it looks like a gene may be expressed, but there are no genes in the centromere. There can't be. That's epigenetically modified so that it cannot translate. But over here, we have all kinds of stuff, and that's what Tomkins is going to point to. It's good. Oh, no, it's actively expressed. Well, if you look at the entire genome, you'll find that this blue line runs across the entire genome. This is just very small pieces of DNA that they've identified with a computer, and they put them on these diagrams. And yeah, it's all bullshit. But he'll come up with something, you know, if he takes the time to look at it. I'm pretty sure he would be afraid to go up against me because I am willing to look at the fucking characters. And I think up until now, has anybody actually looked into it in this detail? I don't know.
I cannot answer that question for you.
Jackson Wheat probably has, and James Downard, and I think I know
there are plenty of PhDs, by the way. I'm sorry for my dog; he's barking because he wants out. There are plenty of PhDs who disagree with Tomkins totally. It's Tomkins is a rare exception to the consensus.
Yep. Well, that's sharp. That's sharp. Okay, that's all we got. That took us an hour and a half. Damn. But my son wanted 10-minute videos.
Well, this was not going to be a 10-minute video. This is a very complicated subject.
Yep.
I want to thank everybody for stopping in. I know Robin.
Yes, thank you.
She asked a ton of questions, and that's what we love. And as well as Sydney and Skeptic Philosophy. Ryan Coltastics dropped in as well. Always love it when we hear from her. And if I skipped your name, I am so sorry, but I'm an old man, and there's a dog yapping in my ear.
So Robin's name is Mike. I didn't know that. Anyway. There can only be one, Robin. There can only be one.
No, he's hitting on or riffing on the Kent Hovind PhD thing. Basically, she suggested we need to buy you a PhD, and there's a trailer park.
Oh, I know. I'm sorry. Oh, okay.
And she said, "Yeah, hello, my name is Mike."
So, I misread that. Okay.
You can never say I would never know where to begin with my dissertation because now you know. Hello, my name is Mike.
It's Mike HSD, high school dropout. I'm proud of it. They weren't teaching me anything. I told them to fuck off. Anyway, okay, we're done.
Goodbye, everybody. And
thank you, everybody. Okay, goodbye, cruel world. We're ending the stream.